Hello everyone, welcome to Pharma. I post videos on pharmacy related subjects explained in a very simple and easy way. If you find my video useful for your studies, don't forget to like and share my video and subscribe to my channel. So today we will discuss on lab diagnostic system. We'll discuss about the introduction to diagnostic system and types, then the details on lab diagnostic system, its advantages, disadvantages, and how computer helps in lab diagnostic tests. So first of all, introduction, that is what is diagnostic. So diagnostic, basically diagnostic in medical field, diagnosis means or diagnostic means to identify disease by examination of various signs and symptoms or various samples from the patient, or by image, by getting a radiological image, so scanning the images, you can say. So mostly the basic point from where the diagnostic begins is the identification or diagnosis of the signs and symptoms. So the basic diagnostic system is the diagnosis of symptoms and signs to identify the disease. When the disease is more complicated, then we further go for various other forms of diagnosis, that is sample diagnosis or image scanning, and so on. So next, various types of diagnostic test. So the various diagnostic tests are clinical diagnostic, the basic one, where the disease identification is based on various signs and symptoms in patient. So doctor or physician generally observe the signs and symptoms. According to that, they try to find out or identify the normal diseases. In case of any complex disease, obviously just a clinical diagnostic will not be sufficient, then we have to go for further other types of diagnostic test. But this one is the basic, like in case of normal viral fever, it is just sufficient to go for clinical diagnostic to identify the cause of fever. So next is laboratory diagnostic. So in case of complicated disease, if mostly the disease is due to some infections or abnormal biochemical level in the body of the patient, we can go for laboratory diagnosis. So what is laboratory diagnosis? It is based on the physical evaluation of various samples which is collected from the patient. So the various chemical evaluation, my mistake, various chemical evaluation of the samples which is collected from the patient. Physical evaluation is the clinical diagnostic test, and the chemical test which is being performed on various samples collected from the patient includes lab diagnostic test or laboratory diagnostic test. Next comes radiological diagnostic. If there is any bone fracture or any or requirement to detect the tumor in any patient, if a physician is guessing that there can be a tumor development, or if there is any fracture, sprain, etc., then there is requirement of radiological diagnostic, which includes medical imaging and its study of the medical images. And then next comes the computer aided diagnostic, which is the advanced form of your radiological diagnostic test, where various sophisticated softwares are being used, that is IT technology, that is the normal radiological system is combined with your IT system or your computer system, by which the medical images which are collected, either it is collected digitally only by the sophisticated software system, and also the images are being scanned by the software only, so that it is more helpful to identify each and every smallest abnormal areas which cannot be physically evaluated by any physician. It is like if a physician is observing or scanning an X-ray report, he may not identify a small breakage or fracture, but when we will go for a computer aided radiological diagnostic, it can identify very easily the minor fractures also. So this is one of the benefit of using computer aided radiological diagnostic. Anyways, we will study about these in details on the next topic. Today we will focus on your laboratory diagnostic test and how computer is helping in this field. So what is basically laboratory diagnostic test? I already told that it is based on the chemical test on the samples collected from the patient. So it is generally used to identify microorganism causing infectious disease and to give its appropriate treatment. It is also used for detection of non-infectious disease in case of abnormal chemical constituent inside our body, like abnormal glucose level, abnormal bilirubin level, abnormal urea, abnormal creatinine, etc. So the most basic parameter is regarded as in clinical test is that it should give proper reliability and validity. That is, whenever we are performing any diagnostic test, we just require two things, that is reliability and validity. Reliability means the result is correct one and it is reproducible type. That is, for example, for like we are conducting a diagnostic test for malaria, and in the first test we found the patient as positive, and then when we are repeating or in any other lab, if the test is again being performed on the sample of the patient, it is again found to be positive. That is a reliable result, that is a correct one. And thus when we get a reliable result, we can say the result is validated. Validated means we can say confidently that the result is the correct one. So reliable result means reproducible type of result, and your validating result means a correct, a result which is actually correct. There is no fault in it. We can confidently say that a patient is actually malaria positive. So this was an example. So anyways, if these two parameters are not met, then the test value is of no use. A physician cannot come to a conclusion that what type of treatment he can give. For example, if a patient is found to be having fever, but the pathological test can't find what problem is there, that why the fever is reoccurring, actually you can say reoccurring. So generally, in the first chance, a physician can say that it is just a common cold and cough fever, or he can say that it is normally a viral fever. But if the fever is going on very severe, it is reoccurring, it is continuously occurring for a week or more, then he will just suggest for a laboratory diagnostic test. And if we are not getting a proper result, then the physician will not able to just conclude that what type of treatment he can give. So this is one example that why we need computer. This is the reason why we need computer, because maybe in pathology there can be a manual mistake. So there can be any unreliable or any false data, it can come. But when we are using a computer system to diagnose or to perform the laboratory diagnostic test, then it will always give a reliable and validate result. So this is the benefit of using computer to why? Because to fulfill these two parameters, reliability and validity. So this is it. Now next, coming to the advantages. So various advantages are that this will give clinicians a rapid right result with appropriate test for doubtful health conditions. And then the weak points in the test processes are readily recognized, necessary changes are done immediately. So if, for example, if a patient is having malaria and we are just testing for any other like dengue, so if the patient is not dengue positive, he is just having malaria, so the test will suddenly show that there is fault in the testing procedure. We have to perform the malaria test. So if we are conducting computer diagnostic test, it will suddenly show that there is wrong procedure being used, and the correct one we can just change as soon as we got the summary that this is negative and all. So for manual test, we again have to put the chemical, we have to go through various types of fever related tests. But in computer diagnostic, maybe the software is so advanced. If the software is that much advanced, it will in its own so that the sample can contain this type of antigens and or this type of pathogens actually, and it can identify that what type of fever is being summarized from that sample. So this is one of the advantage. Then next is that it is human errors are minimized here. There will be no human error, like as I told, while manually doing the test, maybe there are chances that we get a false positive result. Like for example, a patient is having just a normal viral fever, but while diagnostic report we found that he is malaria positive, and the doctor has started giving him malaria treatment, but there is no requirement. At the end we have got to know that there is no requirement, he was just having a viral fever. Or from any, again when the patient is going, or if the patient is again going to any other laboratory, there he is being found to have negative, malaria negative result. So there is no validated result, but when using computer there will be a validated result, so there will be no human error. Now another advantage is that diagnostic decisions are very transparent. Here the diagnostic result is reliable, as I told, and transparent, because it will clearly show what is the problem. It will clearly generate the report of exactly what problem is there, what type of microorganism has been found in the sample, or for example if we are just identifying various biochemical values, like how much glucose level is there, or how much urea level is there, thyroid hormones, etc. It will just clearly show the values. So it will be easy for the people who are taking care of the patient to read thoroughly the report. And the last one is, it is important to detect any outbreak if suspected. For example, in certain local area, a pathological lab is testing on various samples, and he is identified, or he just summarized that out of 10, 8 samples are uh, such samples which are having malaria positive. So from that he can just guess that there is an outbreak of malaria, that is particular area is getting malaria infected. So outbreak can be identified very easily by lab diagnostic test. It can be manually also, it can be with the help of computer also. So lab, it is for common lab diagnostic test that it is helpful in identifying any outbreak. So next, talking about the disadvantages. The disadvantages are not applicable among patients with complex disease. Laboratory diagnostic test is not useful if any patient is having complex types of diseases, that is very complicated, or various types of disease if he is having. So for that we can go for the radiological diagnostic actually. So lack of flexibility and individuality, that is lack in the changes of method. Obviously, for if we want to go for test of urea, since the laboratory test is based on some chemical reactions where the reagent reacts with the sample's biochemical, that is the biochemical present in the sample or the microorganism present in the sample. With that, the reagent reacts and it produces some product. The amount of biochemical or microorganism which is present in the sample decides how much product will be produced. According to that, a value is generated. The more reactant, that is the more biochemical or microorganism present in the sample, will give the more product when it reacts with the reagent. So basically, there is because it is chemical reaction based, so we can't change the type of test according to our need or according to individual patient. Like in images, medical image generation, it is individual, because according to patient's requirement, we can go for CT scan, X-ray, ultrasound, etc. And the images will be the unique one based on the patient's image, which is generated from the patient's body, part of body. But in case of test, the test is common, which is for each and every types of patients. Like for glucose, there is certain type of common test. For blood testing, blood urea testing, there is common type of test, and so on. Then next is only suitable for patients with clear symptoms. If the patient is having a clear symptom, then only we can go for laboratory diagnostic test. That is, we can just make a conclusion that if we identify that maybe he is having diabetes, then we can go for glucose level test. If we are identifying that he is having more urea formation in the body, you can go for urea test, and so on. So but if there is no clear symptoms of any particular type of disease, we have to go for medical image generation to find out that in which part of the body what is the problem actually occurring there. So, and then one more thing is that images is not included. Obviously, it is based on chemical reaction where product is being formed. So we can just know the values of any biochemical or microorganism, but we can't find out the image, that how the microorganism is affecting the certain area, or how the biochemical is harming inside our body. So the next is how computer helps in lab diagnostic. So taking bio samples, analyzing the result is an important part of working in a medical lab, and computers do much of the work there. The sophisticated computer technologies can quickly determine whether the level of protein, amino acids, sugar, etc. are within normal ranges or not. The same computer can also help to generate results that can be sent back to the physician, the patient's physician. So these computers both helps to identify the biomolecules values, and also helps to generate report which can either be printed and hand over to the patient or patient's physician, or otherwise it can be simply mailed, that is from the software system. If there is provision for mailing system, then we can just directly from the software system we can just mail the report to the patient's physician. So now we will discuss about some examples of some digital machines. Like first of all is ABG machine. So ABG is arterial blood gas analyzer machine actually, which identifies the gases amount in the blood. So you can see the image how it looks actually. So the blood gas analysis is also known as arterial blood gas analysis, is a test in which the amount of oxygen and carbon dioxide as well as the acidity, that is pH of the blood, is being measured. So it also helps in measuring the electrolytes and metabolites. So this test can be performed with this digital ABG machine, which you can see. So you have to just put the samples inside these types of digital machine, and the sample will be evaluated. The sample, you have to put the sample, the reagent, and the product which is being formed after reaction between the sample's biomolecule and the reagent will be evaluated and will be given a numerical value form by this digital machines. So next is biochemistry analyzer. An automatic biochemistry analyzer is a medical diagnostic lab equipment which is designed to measure different chemicals and other characteristics in number of biological samples with minimum or minimal human assistance. So it is being used, this analyzer is being used to identify or measure value of various types of biomolecules which is present in the samples. Next is our urine analyzer. Urine analyzer, particularly for analyzing the urine, it is an equipment which is used in the medical laboratory for performing automatic urine testing. So urine analyzer can analyze the bilirubin, protein, glucose, RBC levels in the urine. So you can see this is digital machine where you don't have to go for test tubes and reactions within the test tube or beaker and all. Just you have to put your sample in this analyzer. There will be a sample loading space where you have to load your sample and reagent, and the result of the values will be determined by the analyzing machine, urine analyzer. Next is one more, electrolyte analyzer. So these are used to measure the electrolyte level in the human body, in the sample of human body, and it is used to detect metabolic imbalances, measure renal and cardiac function, as also for blood plasma, serum and urine samples. So for this analyzer we can test electrolyte in both urine as well as blood plasma. So we can test blood also, the serums also, and also we can give urine to identify the electrolyte level in these types of bio samples. It will help us to identify various metabolite imbalances, or if the renal and cardiac functioning is abnormal or normal. Next will be we will discuss on computer aided diagnostic system. So that's for today. If you all find my video useful, then just put a like, and for more videos then go and check my channel. One more thing before ending the video, that is a Hindi explanation of this topic is already being discussed, for which I will add the link in the description box. And if you want notes of this topic, so for that I will add the blog link from where you can go and check the notes, from where you can just simply write the note of these topics. So hope that you have found my video useful. If yes, then press the bell icon to get the notifications. Thanks for watching and bye bye.